Get ready to enter a brave new world with your host, Vasant Dar.
Brave New World is supported by the Center for Data Science, or CDS, at NYU. If your organization is interested in engaging with CDS through student projects, please email cdsindustry@nyu.edu. For more color on the podcast and additional commentary, please subscribe to my newsletter at vasantdar.substack.com. The area of gene editing is on fire. Even before CRISPR, our genetic code has been an intense area of inquiry, but new tools like CRISPR are shining a spotlight on genetic therapy. These tools are powerful enough to create new forms of life, which makes them a cause for concern as well. Helping us make sense of this landscape is Dana Carroll, Distinguished Professor at the University of Utah School of Medicine. He's also a member of the Nuclear Control of Cell Growth and Differentiation program at the Huntsman Cancer Institute. Dana is a pioneer in the space of gene targeting tools and has written extensively about the science and regulatory aspects of gene editing. Dana, welcome to Brave New World. I am delighted to have you on the show.
Thank you for having me, Vasant.
Dana, you've been a pioneer in the space of genomic editing, and you've been writing about its ethical and societal implications as well. Tell us about what got you into the field and your journey.
Yeah, so I'm a professor at University of Utah School of Medicine, and I started my scientific life pretty much as a chemist. I went to undergraduate and graduate school in chemistry, but was kind of unsatisfied with what one could do in chemistry. I felt the need for something a little broader. So when I was a graduate student, I attended a lot of seminars on biological topics and ended up, after I finished my PhD, going off and doing a couple of postdocs. And I was very, very fortunate that in my second postdoc, the tools of DNA manipulation were just getting started. You know, the ability to cut DNA with a restriction enzyme and sort of get deeper into what's going on with DNA sequence, the ability to make molecular clones of DNA, eventually DNA sequencing. And so I kind of grew up in molecular biology with the tools as they were getting more and more powerful. And I had been studying how cells deal with damage to their DNA in my own research for a while. And that's what led me into getting interested in how to change DNA inside cells. And so that was sort of the beginning of what we now refer to as genome editing, where you can actually go in and make very specific targeted changes in DNA inside cells and inside organisms. And this is go ahead.
No, I'm sorry to interrupt, but I just can't help but asking this. When did the larger implications of this work start becoming apparent to you? You know, the fact that in principle, you could now design life, right? Something that was just, Frankenstein and science fiction, right? Like, when did that strike you, the sort of the implications of this?
The discussions about that happened long before genome editing was even available. Once we were able to isolate individual genes from the total DNA of an organism, people began talking about gene therapy. So if you have a deficit in a particular gene, and I'm able to isolate from somebody else's DNA a normal copy of that gene, why can't I give that normal copy to you, right? And so even just when we had molecular cloning back in the 1970s and 1980s, people were already talking about how to use this technology, and could we actually make permanent changes in people's own DNA. And that discussion got more urgent, I would say, with the development of genome editing.
Very interesting. So you've talked about having grown up with the field as the tools became more sophisticated. And now we have CRISPR where we can edit gene sequences. Let's define a sequence because DNA is a double helix and yet a sequence is one strand of it. So we assume that the other strand is implicit?
The DNA inside the cell is a double helix, but each strand of that helix contains the same information content because if there's an A on one strand, there's a T on the other. If there's a G on one strand, there's a C on the other. And that's throughout the whole length of the DNA. So inside one of our cells, we have 6 billion of these base pairs. We call the A's, G's, T's, and C's bases, and they're in these ATGC pairs. And we have 6 billion of them. But because each strand has the same information content, you copy a strand that has to be copied with a G in the other strand. So if we sequence one strand, which is how it's done, we get the whole story.
Yeah. And that's so obvious now that you state it that way. But I did wonder about that. So I guess when I read Jennifer Doudna's paper, that was sort of the first time I'd come across this notion of sort of programmability. That you could actually program how you wanted to edit the DNA. And I'd always sort of thought of programs as software. So that was like news to me that we can actually program things to make changes to our genetic structure. How does that happen? Like, what does the biological programming consist of?
So the tools of genome editing can be programmed to go to a unique site in all of the DNA in the genome of any cell and make a change there. So that's the power of the genome editing technologies. They can be programmed, directed to go to a unique site, and you can change your mind about which site they go to.
Now, how does that happen? Does that happen? So in her paper, she talks about sort of the RNA molecule as a guide. So say more about that.
Right. So people often make the analogy with a text editor. So you've got a cursor that you can place anywhere you want in text. And then from that point, you can begin to make additions and deletions and replacements in the text. And the RNA in the CRISPR system gives you the ability to place that cursor where you want to. So let me just say there are two fundamental pieces to the CRISPR system. One is a protein that's called Cas9. Now, this protein can make a break in DNA, but it can only make a break in DNA when it has a small RNA molecule that can tell it where to go to make that break. And so that RNA is the key element. It has the information that tells Cas9 where to make a break in DNA. Now, you might think, make a break in DNA, that sounds like a really bad idea. But this is where the studies of how cells repair breaks in DNA come in, because cells have multiple ways that they can repair breaks in DNA, and we take advantage of them to make these insertions, deletions, and replacements at the point where the RNA has told Cas9 where to go.
That's just fascinating, and thanks for expressing it so clearly. So what were previous attempts like at gene editing, and what were their flaws?
Well, the first of the tools that we would call a gene editor was something called zinc finger nucleases. And these were proteins that were made up of a part of the protein that can cut DNA, but it doesn't have any specificity. And then linked to that was another part of the protein that could direct the cutting part to a particular place in DNA, like the RNA does for CRISPR. Okay? But this was all protein. And the recognition part of that protein could be varied, could be altered to take the cutter to different places in the DNA inside a cell. So it had the same capabilities of CRISPR, but it was more unwieldy because you had to make a new protein every time. And the modules in that protein that led to DNA recognition weren't always great. So
So in terms of the analogy to the cursor in the book, if you sort of stretch that, are you saying that it just took a long time to get the cursor to the right place, and sometimes it wouldn't quite get to the right place?
Yes, and just designing the cursor to get it to the right place. So the wonderful thing about CRISPR is that it's a pretty small RNA molecule that provides that specificity. And you can just redesign an RNA. They're very easy to make, these RNA molecules. And so you can go pretty much anywhere you want in the whole genome full of DNA to a unique site and make those changes.
Fascinating. Doudna's article also talks about makes reference to this genome manipulation toolbox. So I guess what I'm hearing is that this is one of those tools in the toolbox, the Cas9. What other kinds of tools are there in this toolbox that she's talking about?
Well, what people have done, and they've done this both with some of the previous platforms, but also very effectively with CRISPR, is to take the recognition capabilities of the Cas9 molecule with the RNA and put different activities onto Cas9. So let's say we don't want to make a break in the DNA. We want to come in to use the recognition capabilities, go to a unique site, and just change one base. Okay? There's a T there. We want to make it a C. So we come in. We make a Cas9 protein that can no longer cut DNA, but we stick onto it another piece of protein that can do this base change, can just change. The example I picked was kind of a bad one. If we want to change a C into a T, there's an enzyme that will do that in DNA. And so we put that onto the Cas9 protein, which can no longer cut DNA, but now it has the ability just to change that one base. And we use the specificity provided by the RNA.
Wow, that is so cool. So it changes. So I guess it changes the base pair, right? Because if it changes one, it has to change the other one as well. Wow, that is just so cool. So, when I went back and sort of looked at the history of, the Human Genome Project, I guess when it got started, it used to be excruciatingly slow, right? To do the sequencing. So just summarize for us, what turbocharged this process? What did computers really enable us to do that we weren't able to do previously?
Started working with lots of different technologies to try to build an approach that would allow doing high throughput sequencing, doing lots and lots of different parts of the whole genome simultaneously. So you've got 6 billion base pairs. You can cut it up into little fragments and use an approach to determine the sequence of each of those little fragments. And once you have those sequences, as long as you have enough fragments covering the genome and they overlap with each other, you can reconstruct the whole genome sequence just using a computer, right? But you have to get these millions upon millions of individual sequences. So people started working on these technologies, and there were, 6 or 8 different technologies that people were working on through the 1990s and into the early 2000s. And a few of them have now taken over. There's a company called Illumina It's in the San Diego area that has kind of become the go-to commercial outfit for doing massive parallel sequencing. But there are other approaches, other companies and other laboratories that have approaches to DNA sequencing. But a lot of it had to do with things that are happening in all sorts of areas of science, just doing things at a very, very high throughput level.
Interesting. So I guess when you do things at that scale, you also make mistakes, right? So occasionally, I guess these machines would make mistakes. Have we gotten to the point now where we do this thing basically without error, or are there still potential errors in there?
We iterate, Vasant. You don't just read through once. When you fragment the genome to make these individual sequences, you'll have since you have DNA from a lot of cells when you start, you've got many copies of the same DNA sequence, and you sequence a bunch of them. So you have read what should be the sequence multiple, maybe thousands of times, and you take the best read at every position.
Got it. So you reduce that epsilon error. You get that as close to zero as possible. But okay, that's awesome. So one of the things I was wondering about, when I started reading about the technology, is what role does artificial intelligence play in any of this? Where does it come in? Where do smarts come into the picture?
I don't work on AI. It's being used, as I understand it, mostly for trying to develop predictions. So if you're trying to figure out what's responsible for a particular genetic disease, you can use elements of AI to try to look at a lot of DNA sequences, and where you don't have direct evidence, you can try to look at similarities with other sequences that have similar effects. It's also being used in structure prediction for proteins. And I just was reading something the other day about another approach using AI to just take a sequence, a DNA sequence of a gene, which will predict the amino acid sequence of a protein, and then just go from that sequence of amino acids to try to predict in 3 dimensions what that protein will look like. And
very, very interesting. Yeah, I was aware of that work. In fact, a couple of my colleagues are doing exactly that, and they're working, I think, with one of the large companies out in the Bay Area on this sort of structure prediction. I guess predicting folding properties and things like that. More generally, it looks like maybe the role of AI, as you say, is prediction and in sort of enhancing the understanding of what these genes do. Because my understanding of the field is that we still are sort of at the early stages of trying to understand, what large sections of this DNA actually do.
It's true. So we won't know until we test it experimentally. I'm an experimentalist, but
right.
These prediction schemes have the ability to set us on the right track. One of the things that one of my colleagues is doing is trying to look at how genetics influences metabolism. So I tend to think of DNA is over here, and what we eat and how it's processed in our body is somewhere else. But of course, the genes that are embedded in genomic DNA are governing how our metabolism operates. And so it's been difficult, since metabolism is very complicated, difficult to know exactly how particular genes are influencing different aspects of metabolism. And so we can begin to get some sense of that by looking at things that change in metabolism when a gene has been altered, and looking to see what the influence is rather broadly. And then we can try to go back and see what pathways, genetic pathways, regulatory pathways and metabolic pathways could be responsible for these changes.
Very interesting. You mentioned testing, and I was thinking about that, in preparation for our conversation, that testing has got to be like a really thorny issue here, isn't it? I mean, I was thinking of the Chinese scientist who, I guess, altered some genes of these twins to make them HIV resistant. And I was watching this conversation between Jennifer Doudna and Chris Anderson, and in that conversation, he asked her, like, how would you actually test whether it works? Would you actually expose these babies to HIV to test whether this thing works? And in reading, one of your articles, I also noted that, you talked about the CCR5 knockout gene and the fact that, if I remember correctly, that the evidence was that it wasn't 100% effective. That in reality it wasn't what they expected. So I guess the larger question is, like, how the hell do we test something like this?
Yeah. So CCR5, the one that J.K. He, this Chinese scientist, tried to did modify, as far as we know, in these children, that was one that had a lot of good evidence in humans already. There are some natural human mutants that lack CCR5, and we didn't know that those people existed until the AIDS epidemic. And some of these people began coming into AIDS clinics. They were HIV positive, but they never progressed to AIDS. And it turned out that since CCR5 is required for most strains of HIV to enter T cells, that their T cells were protected from being killed by the virus. So although they had the virus in their system, their T cells were resistant. Those are natural human mutants. They're walking around the streets. You wouldn't know who they were unless you did some genetic testing. So that was actually a good choice for a target. So there are lots of issues around this, Vasant. I'll try to limit what I say about it. But right now, we do not know the condition of those girls. I was convinced, after looking very thoroughly into this case, that J.K. He actually did what he said he did, that the children were born. He had data to show on cells from them after they were born. So I think he did what he said he did, but we don't know anything about them. One of the very unfortunate things about this whole business is that only one of those girls had both alleles of CCR5 modified. So as you know, we get for each chromosome, we get one copy from mom, one copy from dad. So we have two copies of what are called alleles of any gene. In one of the children, both alleles were modified, so they shouldn't produce any CCR5 protein. In the other child, only one was modified and the other was still good. So she would not be protected from HIV. And that's one of the terrible things I think about this whole story, is that he went ahead and implanted an embryo that he knew from embryo testing would not be resistant to AIDS.
So was that by design, or was that an accident?
So,
or just chance?
It was chance. So what he did, he was working with some in vitro fertilization people. It must have been in an IVF clinic. And so after the in vitro fertilization had taken place, the standard procedure is to keep that fertilized egg in the laboratory and monitor its development for a few days, just to make sure everything's going okay, before implanting that embryo back into mom and letting you can't get complete development outside of a mother. So it has to be implanted back into mom, where it will go through the rest of development. But before that, those two embryos were implanted, he was able to take a very small number of cells, like 3 cells, and specifically ask by DNA sequencing, have both alleles of CCR5 been modified? And so he knew in one of the embryos by DNA sequencing, both had. In the other embryo, only one had. And it's the thing about CRISPR is he injected the CRISPR components into the fertilized egg, one cell stage. But it's just, CRISPR is not 100% efficient. So you're just sort of relying on it being as efficient as it can be. There are 2 targets in each cell. Sometimes you get one of them, sometimes you get both, sometimes you get none, for reasons that are sort of stochastic.
Interesting. So this makes my question, I guess, even more pertinent, which is that, the testing just seems like it's going to be a difficult thing to do, isn't it?
So this is something that's done in IVF clinics around the world on a fairly routine basis. It's not done every time because most IVF situations are ones where you're just trying to overcome an infertility. But in some cases, there's also a disease gene that may be segregating in the family. So you want to make sure that the embryo that you implant isn't going to carry that disease. So let's think of 2 parents, both of whom carry a cystic fibrosis mutation. So they have one good copy, one bad copy, each of the parents.
Mm-hmm.
So when they fertilize a bunch of eggs, each egg has a 25% chance of not having any bad copy, 50% chance of having one bad copy, 25% chance of having 2, which would lead the child ultimately to have cystic fibrosis. So you can do this genetic testing on the embryos after in vitro fertilization and make sure that you're implanting ones that don't have 2 bad copies.
I get that, but by testing, I didn't mean that as much as does it actually work?
All right. So for knocking out CCR5 for HIV, as I said, there are very good human precedents. The prediction is very strong that the child who has the 2 knockouts will be protected from most strains of HIV. It's also true that there are some strains of HIV that do not depend on CCR5, but that's a separate issue. So I would say the prediction is so strong in that case that I don't think a further test is necessary. This is true for cystic fibrosis also. If we screen the embryos, we find ones that don't have a bad copy. By implanting those embryos, we will be quite confident that they will not suffer from cystic fibrosis. So what's the key here? The key is that each of these conditions is mediated by one gene, and we know that. Okay, there are quite a lot of single gene diseases, and we know for a fact that just by looking at the segregation of the bad copies of the genes and the disease phenotype, we know that. What's not clear is that if somebody tries to modify a gene where we don't know that that's the sole cause of a condition of some kind, will that have the desired effect? So, this is, we can jump ahead, if you like, to the idea that people might say, well, if you can change a gene to protect somebody from HIV, why can't we change a gene to make my kid smarter? And there are a few genes out there that people have shown to have a very, very small influence on intelligence. So this is in quotes, intelligence. Yeah. So you tell me, what's intelligence?
I've been trying to figure that out for a long time, Dana.
Right.
Indeed. Yeah.
So The problem there is, first of all, the measures of intelligence are faulty. Everybody uses surrogates of some kind for measures of intelligence. Second thing is that the genes that have apparently been identified to have some influence on intelligence rely on those surrogates, but also individually through large-scale genetic analysis have only a very, very small effect on those surrogate measures. So we don't know how to measure intelligence. We certainly don't know how to change it. And so you go into something like that and you're completely in the dark.
Thanks for that, Dana. So I guess as long as we stick to single gene editing therapies to treat things like cystic fibrosis and Alzheimer's, the predictions on outcomes are pretty solid compared to sort of the nebulous things like intelligence, which gets into ethics and all of that, which I'll come back to. But switching gears a little bit, is there anything going on in the use of CRISPR for the creation of new kinds of foods?
Absolutely. I think the impact of CRISPR genome editing in general on agriculture is going to be much broader than the impact in medicine in the next 10 years, possibly longer. So let me give you an example. A friend of mine, Dan Voitas, started a company in Minnesota to begin to modify crop plants to give them better characteristics. The company has been working on better nutritional value. They're interested in disease resistance, things like that. And they actually have a product on the market. You can buy it. It's a soy oil that has been modified to have healthier characteristics. So soy oil, like a lot of plant oils, has quite a lot of unsaturated fats, polyunsaturated fats in it. So what they did was they modified in the genome of the soy plant genes that led to the production of these polyunsaturated fats, particularly in the bean. So now they only make monounsaturated fats. They don't make the polyunsaturated fats, and the fat characteristics are more like olive oil, which is considered to be very healthy. And so now when oil is produced from these soy plants, it has that characteristic. And it was just by knocking out a few genes using, they were actually using a previous genome editing tool called Talons, but it does the same thing that CRISPR does. So that's an example, making a healthier soy oil. People are working in both livestock and crops to make organisms that are more resistant to disease, but also more resistant to climate change. So there's an example, there's a company that I used to work for on their scientific advisory board, again in Minnesota, that's working on trying to make cattle more resistant to high temperatures. So in dairy cattle, they're very sensitive, as I understand it, to increases in temperature. And, milk production goes down, their general health is compromised by high climate temperatures. But there are breeds of cattle, particularly in Puerto Rico, that are comfortable in much higher temperatures. And it's due, at least in part, to a single gene that's called SLIC. And so if you can make the SLIC modification in dairy cattle, you can make them more resistant to increasing temperatures.
That's interesting. So why does GMO get such a bad rap? Is this justified? Like, is it overreaction? Like, what's your thinking on this?
I think it's the key problem is that the first GMOs, among the first GMOs that were rolled out, were the herbicide-resistant crops that Monsanto produced. And Monsanto produced the genetically modified crops. They produced the herbicide that they were resistant to, so you could kill weeds but not the crops. And they sold both the seed and the herbicide. So it was a clear example of corporate greed for many people, So I think that's what got a bad reputation initially for these genetically modified crops. There isn't any evidence that the genetic modifications that I'm aware of in crops like corn and cotton and soy that are being used as livestock feed, and some of them, of course, are going into human food. There's no evidence that the genetic modifications per se have any adverse influences on human or animal health. So it got kind of muddied for what I think is a legitimate reason. So my thought experiment is, what if the first genetically modified crop was a drought-resistant cassava that was given essentially for free to farmers in sub-Saharan Africa?
Yeah, no, exactly. Completely different story, different positioning, and probably would have resulted in very different views of GMO, indeed.
So let me just say one further thing. So the great thing about plants and animals, crop plants and livestock, is that once you have an example of the modification you want to make, and you've thoroughly characterized it, which is really easy. You can do whole genome sequencing. You can do very thorough characterization of the plant or the animal. You can test its health. You can test its productivity. You can feed it to mice. You can feed it to people, on a small scale, sort of go through all the testing. And then once you've decided that this is really a beneficial genetic change, you can spread that extremely rapidly through seed and semen. This is not so possible. This is not so possible with people.
Indeed. Talking about animals, one of the approaches people are pursuing, and I believe they haven't made much progress yet, is this notion of cellular meat, like real meat, making meat from cells. Is that like an area where genetic technology could work in just producing artificial meat, like real meat?
I think it's probably an issue of scale, because to grow whole plants is pretty cheap, and you can scale it up. But to grow lots and lots of cells, you've got to feed the cells something. They have to be in a sterile environment. You can't really take it out of the laboratory and plant it in the ground. So to make cellular meat, I think, is going to be hard to scale up. I don't pay a lot of attention to that area. I do eat some of these plant-based meat substitutes. I find them just fine substitutes, although my wife doesn't like them. So
yeah, they're quite good. I mean, I tried them for taste. I mean, I gave up meat several months ago. I read this book by Peter Singer called Animal Liberation. I just that changed how I've looked at sort of animal farming. But yeah, they are quite amazing. So in both in terms of texture and taste. So I want to talk a little bit about sort of this germline editing, which seems to be something that's incredibly powerful and whose implications we don't really understand yet. So say more about this. What is germline editing, and what are sort of some of the open issues around it?
Germline editing is what J.K. Hood did with these CCR5 girls. He had access to embryos at the one-cell stage. He injected CRISPR. CRISPR did its job more or less. The embryo was implanted and children were born. Now, those children carry mutations. They carry those modifications in every cell of their body, because the modifications were induced by CRISPR at the one cell stage. So when that cell divided and every other cell, as the embryo grew and then the fetus grew and as a child grows, every cell carries that modification. So the germline is sperm in males, eggs in females. Those are the cells that we pass on our inheritance to. And so amongst all the cells in those girls' bodies, all the eggs they produce during their lifetime will carry those modifications. So that's what germline genome editing is. You're making a modification that happens early enough in development that it's going to be carried by every cell in the body and has the potential of being passed on, if the person reproduces, will be passed on to subsequent generations.
So as another example, if there's a gene that's responsible for high cholesterol, you can, I guess, make the edit so that it's not passed down, which is not germline, versus one where I guess the program for it's almost like the software is passed on as well, isn't it?
Right. Right. Yeah. So there is such a gene, and people have been working on a therapy for grown people to try to knock that out in particular cells in the body to reduce the production of cholesterol. And
so are we like potentially playing with fire here? I mean, I can imagine, let's say, the traditional approach to lowering cholesterol, which is statins. I don't know what side effects that has, versus the genetic approach, which is one shot, boom, it's gone. It's gone from all your descendants. But I guess, what if I have that high cholesterol gene for a reason? I mean, there are, I guess, regions in the world where people have high cholesterol. I have high cholesterol, and it's genetic. But touch wood, no one from my family has had heart problems, which are typically associated with that. So I guess I'll come back to my question, which is, maybe things are the way they are for a reason. And when you mess with one part of your system, there are all these sort of Rumsfeldian unknown unknowns just waiting to, happen.
Absolutely. That's a really good insight. It's true that the functioning of our cells and our bodies is very complex. And you tweak one part of the system, you take out a spark plug from your car, what's the effect of that going to be? And the gene that people are concerned about for cholesterol, high cholesterol, is not one that you could take out altogether, as far as I understand. That would really mess up the system. We talked before about CCR5. That's one where at least some people do fine without it. But there are subtleties. So one of my concerns about these Chinese girls who were born is that the lack of CCR5 is practically unknown in Asian populations. It's characteristic of some people in Northern European populations. Over centuries, the absence of CCR5 may have gone along with some adaptations elsewhere in the genome to allow cells to get by without CCR5. CCR5 is present in all mammals. It's not something that just turned up sort of accidentally in people. So it actually has a function to perform. Knocking it out in a situation where we don't know that that's going to be allowed physiologically seems to me like a really bad idea. So, and this is true of essentially every germline modification that you might consider. And that's partly because, although we can identify a single gene defect in a child or an adult as being due to a single gene, we don't know whether there are effects prior to birth that are dependent on versions of a particular gene that we don't understand. We don't understand much about how genes operate during human development, the very earliest stages of human development. So making modifications that are going to be present through embryo development and fetal development on into child and adult development, it's a very, very tricky business.
So what do you think the regulatory path is going to look like here in this space? Are there certain things that seem safe where one might even say, well, it's irresponsible not to do it? So are there sort of no-brainer kinds of applications like that versus riskier things? I mean, how are regulators thinking about this space?
Yeah, there are a couple of reports that have come out in the last couple of years that I can alert your audience to. I was an author on one of these reports generated by the UK Royal Society, US National Academies of Sciences and Medicine. And we looked at the technical aspects of germline genome editing. That report was published by the National Academies of Sciences in 2020. And there was a World Health Organization report that came out last year that I think you can get for free from the WHO that was more talking about the societal and regulatory aspects. So I have a lot to say about this. Let me try to say a few things quickly. Okay?
Yeah. No, take your time. Yeah, no, because I think this is a fascinating open area. So yeah, let's talk about it.
I do too. I think it's very important. So the first thing that our report said was, if you're considering doing human germline genome editing, at least in the first instances, you should only do it in cases where the disease modification in a single gene has been very clearly identified, and it is absolutely clear from all we know about the biology of that system that making the change you want to make will avoid that condition, that disease, whatever it happens to be. So sickle cell disease is a very good example of that. So let's talk about sickle cell disease. The second criterion, before you do even the first attempts, would be that the only people who should be subjected to the germline editing would be couples who have no alternative for having a genetically related child without the disease. So let's imagine a couple where both mom and dad are sickle cell sufferers. They both have 2 copies of the sickle cell allele, and they are sickle cell sufferers. When they have children, their children are all going to get 2 copies. They're all going to have sickle cell disease. But if we did in vitro fertilization for this couple and used CRISPR to modify those embryos, monitor the embryos to make sure our modifications worked as designed, and then implanted those embryos, the children would no longer have sickle cell disease. Make sense?
Yep, makes total sense. Yeah.
Yeah. So that's the first 2 things. You got to have a good handle on the genetics, and you only use it for people who have no alternative for having a genetically related child. Third thing is, CRISPR can't work sufficiently efficiently and accurately to do this now. The technology just isn't up to it. Just like we saw with the Chinese CRISPR babies, only one of them had both alleles modified. The other one was still going to be susceptible to AIDS. The other thing that's a worry here is CRISPR's very good at finding its target and making modifications that you want to make at the target, but it has a low level of going off and making changes elsewhere in the genome. It is a low level. For many applications, you can completely ignore that or take account of it in pretty straightforward ways. But in embryos, you've got no chance to go back. You've made that change. What do you do next if it's in some other gene? Okay, so maybe you see that you did just what you wanted to do in the target gene, but there's something else you did inadvertently at the same time. So first of all, the technology has to be specific enough so that it will only make a change at the target. And second, it has to do that efficiently enough so that you're confident that you're going to be hitting both alleles. So we aren't there yet with the technology. So there has to be research that will get us to that point. The third thing that I think is really important is society's not really ready for this yet. And so both our report on a sort of smaller scale and the WHO report and a previous report from the National Academies emphasized the need to get the public involved in thinking about whether we want to go ahead with this. And then finally, there's this issue that's related to things like trying to improve intelligence. And people have lumped this into the category of enhancements. Instead of correcting a disease mutation, are you going to be trying to enhance people's mental or physical abilities or appearance in some way? There's a word for this that I can't come up with right now. And related to that, and actually going back all the way back to any uses of this technology in medicine, are we just going to be creating an opportunity for wealthy people in wealthy nations? Is any of this technology going to be available to the people who need it most? Sickle cell sufferers in the US, of whom there are lots, 100,000 roughly is the estimate, but almost all the sickle cell sufferers in the world are in sub-Saharan Africa and tropical regions of South Asia. So how do you get a complicated, expensive technology to those people? With crops and livestock, as I was saying earlier, that seems like it would be feasible. You've got seed, you've got semen that you can transport into any region in the world. But when you're talking about these treatments of humans, that's going to be almost as far forward as I can see. That's always going to be complicated and expensive.
Yeah, I noted that in one of the articles you wrote, where you point out that the costs of some of these therapies can be easily $1 million, and in some cases, tens of millions of dollars, right? So they're quite expensive. I mean, are developing countries also at a comparative advantage in being able to do clinical trials because it's cheaper to do them there? Is that likely to happen? I mean, I'm not talking about China where it's different, but let's take, India or other countries, right, where you have sort of the science is, permeates relatively quickly across the community, and then the economics are more favorable there. Do you see that happening potentially?
I think some of that goes on already. Clinical trials being taken to places where it would be less expensive to run them. But there's also this issue I know less about, but I've been introduced to it, the issue of exploitation. Even if you're thinking about a small molecule drug or something, if I decide I'm going to do my trial, take an area of India where you won't have to pay the subjects very much. The clinic clinic isn't going to charge very much, but the drug turns out to have serious side effects. Haven't we just exploited that population to learn to protect people in the U.S. and France? So I think it's a complicated issue. And
yeah. I can see that. I can see it being complicated. So a related question from your article. So do you think there's a risk that we're going to sort of, I guess, develop a class structure here where, people with means are able to use the technology and others just have to wait till costs come down? Is that the way you see things
absolutely
unfolding?
I think it's a real risk. can we talk about the somatic therapies, the ones that are actually done on, not on embryos, but on living persons first?
Yeah.
Let's talk about that first.
Yes, I wanted to come to that as well. Yeah, please.
I'd like to go back to the germline ones in a minute, but so thinking about these genetic therapies, it's almost inconceivable that they could be transported to developing countries that don't have the infrastructure to support them. Because, talk about sickle cell disease, there are clinical trials going on with sickle cell patients currently, and they are looking very promising. So this is a good thing. But the way the trial goes is you have a patient, the patient is suffering from sickle cell disease. You make that patient anemic in order to get some blood stem cells. They're called hematopoietic stem cells. Okay, so they're blood stem cells. The cells in your blood are turning over throughout your life at actually a pretty fast rate. And so these stem cells, which are normally in your bone marrow, are producing new blood cells all the time. So by making a person anemic, we can get some of those stem cells to come out into the circulation. And so we can collect them from the circulation and bring them into the laboratory. And those stem cells are then treated in the laboratory with CRISPR to make the modification of the gene that has the sickle cell mutation. Those cells are then expanded in the laboratory. They're grown in the laboratory, so there are more of them. They're characterized very thoroughly to make sure there aren't any of these off-target effects, any other modifications that might have been made. Patients brought back into the hospital. The patient is treated like you would treat a bone marrow transplant patient, where you treat them with a drug that is toxic, particularly to the bone marrow. Why do you do this? Because you want those stem cells you produced in the laboratory to go back into the bone marrow. So you have to make room for them in the patient's marrow. The patient is currently making sickle cell red blood cells. You want the number of those to be suppressed while you put in cells that will produce normal red blood cells. Then you infuse the treated cells, the modified cells, into the patient. You make sure the patient is doing okay, monitor them in the hospital, then you send them home. So this takes, I would guess, a couple of months at least for all those steps to occur, many of which are being done in hospitals or in, clinical laboratories. And so there's no way that all of those steps can be done at low cost or in remote regions of the world. So how could you get around? Well, the Gates Foundation at one point was making an investment in technologies that might get around this. And I honestly don't know the status of that right now. But what you'd like to do is you'd like to have a single injection that you could give to a sickle cell sufferer that would make this modification in their own bone marrow. So this is a little bit of science fantasy here. We have the CRISPR components. There's a Cas9 protein and that RNA that we call a guide RNA, because it's going to guide us to the right site. And maybe you have something else in there that's going to help with making that modification. And we put those components into a little package that is going to be injected into the patient. And it's going to know how to go to the bone marrow, find these stem cells, the hematopoietic stem cells, and get into them and allow release the CRISPR reagents and allow them to do their stuff at high efficiency. All right. So that's what we need. There are a few steps in there.
I noticed. Yeah.
So, but to me, maybe ultimately it won't be CRISPR. Maybe it'll be some other technology that is developed that I can't imagine now. But each of the steps in that procedure is kind of a fantasy at the moment, but at least it defines a hurdle that has to be gotten over.
Yeah, I mean, I can see the trajectory that's required to get us there. So, Dana, you mentioned, I guess, the word risks a little while ago, and I want to talk about that here, and especially the risks of, I guess, people just going rogue with this technology, right? Because it seems to me that this technology is accessible to, let's say, a university freshman. Maybe not high school, right? But someone at a university has the tools and the capability at their fingertips to, design life, to be quite grand about it, or to design chaos. In that conversation of Chris Anderson and Jennifer Doudna, I was actually surprised by her response, because he did ask her the question, and she said that, she's more concerned about sort of computer hackers than, let's call these people genetic hackers. And I was actually shocked to hear that, because, I mean, I'm a computer person and I know there are breaches and all that kind of stuff all the time. But we also have people looking out for this stuff, right? We have people constantly designing sort of against or to thwart these kinds of attempts. It seems to me that the same sort of checks and balances or constraints don't exist at the moment with this technology. That, it's the equivalent of like a biological weapon. I mean, I can't put it any more starkly than that, right? That we've sort of given access to everyone. And yet we seem to be kind of driving on as if this threat doesn't exist. Or am I exaggerating this?
You are exaggerating a bit, but let's talk about it. So, first of all, for the clinical therapies that people are working on now, as I said, to get those into patients is actually complicated and expensive. And the university freshman or the person on the street doesn't have access to those or the funding. So it could be that Elon Musk could set up a clinic where he offered to make CRISPR treatments for people. If he has the funding to set up a medical clinic, and he has funding to have researchers who would come in and develop CRISPR for various things, he's capable of doing it. But short of billionaires, it isn't really accessible to people. The germline therapy, germline modification, on the other hand, is much more accessible. So I told you how J.K. Ho went about this. He just allied himself with an in vitro fertilization clinic. And the only thing that he did that was different from what that clinic would normally have done was take CRISPR reagents that he had developed and injected them into embryos. So the person on the street could potentially develop novel CRISPR materials, could potentially formulate them, put them in a really, really tiny injection needle, and get an in vitro fertilization clinic to do the injection for them. So that is much more accessible. The cost of doing that is minuscule compared to the cost of these clinical therapies, where you actually need a clinic. So I'm much more worried about that than I am about the things that are more complicated and work on children or adults. And I think it's a genuine concern. And in fact, J.K. Ho himself did this outside the bounds of the normal scientific and regulatory processes. So people are very sensitized to this now in the scientific community and to some extent in the regulatory community, but not, I would say, in governments and in the general population. And we may be fortunate that the US government, for example, is tying itself in knots over other issues and isn't really paying attention to germline editing or genome editing at all, because they would probably make a mess of it if they tried to regulate it. But we do need to have these conversations, do need to get a sense of what the public is up for and what the public is not up for, and to try to get sensible ethical and regulatory perspectives on this, ultimately get good regulatory enforcement in the long run. But there are going to be places around the world where regulation and enforcement are not strong, and somebody who wanted to put these modifications on the market for people who had money would certainly be able to do it. And that's my real worry, that there are going to be rogue operators that are going to go out there and do this, and they're going to make a mess of it. The next time this is done, it's probably going to be a much worse mess than what happened in China.
Dana, it's been a fascinating conversation. I just learned so much in trying to prepare for this conversation, and even more in talking to you. So thanks so much for sharing your time and making this accessible to regular people like me. Well,
it's my pleasure. The way I often say it is, you put a nickel in me, that's what you get. I love to talk about these issues. I think they're very important. I've been very, very fortunate to be engaged in the many aspects of genome editing for much of my scientific life.
Yeah, fascinating stuff. Great area to be in. I think if I was starting again, I might choose to try, marrying computing and genomics. It seems to be sort of an important part of our lives going forward.
Yeah. We can talk about that offline a little if you want.
Yeah.
One of the things about using informatics is that they tend to make a big deal out of finding little effects, and it's not absolutely clear how useful that's going to be in the long run. I mentioned it briefly in terms of trying to enhance intelligence.
Yeah.
So what do you do with an effect that is, the effect, as far as we can tell, based on sort of faulty definitions of intelligence, has an effect that's below the 1% level, and we don't know what the function is that's related to that effect. What are we going to do with that information?
Yeah, indeed. By the way, one of the things I just remembered, also something I came across in preparing for this, someone said that computers and genomics are ideal. They sort of work in, with discrete data, that computers have zeros and ones, whereas genomics have like a 4-letter, primitive, but they both seem sort of discrete and digital in a sense. So they're well suited. And that sort of really resonated with me. It makes a lot of sense that you would get that kind of precision, when you marry computing and genomics.
And I think that may be why I like DNA so much, is that there is this sort of digital coding relationship that's embedded in DNA. And the problem is when you put the DNA into a biological system, everything gets complicated.
True that, as they would say in The Wire. Well, Dana, thank you so much for that again. It's been a pleasure talking to you, and I will follow up for some literature, especially on sort of the ethical aspects and how the regulator is looking at the space. But thanks so much again. It's been wonderful to have.
My pleasure.
Wonderful to have you. Thanks.
My pleasure.